I'm wrong, and I'm starting the recording. So, and let's see. Okay, yeah. And if you wouldn't mind muting yourself, so that way somebody else who has a different audio system does not get the echo. Okay, yeah. I think yeah, Mike and Ed, you have to mute. Yeah, just a little bit. Or otherwise, I can do it. Yeah.
I was
Okay.
going to suggest you sit back in the chair, lean back a little bit in the chair. And now as you
Okay.
talk, it will be a little clearer to the microphone. Just try it.
Okay.
Yes.
All right. Okay. Yeah. Oh no, no. I wasn't getting an echo. I was just getting. I didn't know if I was
No
fine.
sound and echo, just a little reverberation.
Reverberation. Okay. All right. Yeah. Thank you. I'll I think, yeah, because yeah, I think I'm with my short height on this. So this is the class is entitled Genes, Genome, and CRISPR. So I had did a class on genomics at one time, but then I think either Kevin or somebody else had said, with the CRISPR and the new book that came out by David McCullough, who is almost like a homage to Jennifer Doudna, who got the Nobel Prize for CRISPR. So he had, said, well, we should do a class on CRISPR because there's a lot of, political and East Coast, West Coast competition. So it's almost looked like a clock and dagger story. So that's why I decided to. And this is such a hot area that every month there's a new information. So that's why I decided to do an updated version. So even if somebody had taken it or knows about it, there should be enough. And I'm trying to make it. And also at the Rich Kennedy's discussion, there was a class about the ethics. So I threw in some material because there is seriously some, concern as to how all this, and especially with the Chinese scientist who tried to use CRISPR to modify what is called germline, and that was, almost, and he was sloppy, to put it mildly, and premature, so that really has caused some huge vibes across the world as to how this technology could be used or abused. So it's an interesting story as to how, again, the desire to be the first in everything that caught up with him, and also how what the government's roles were, and so that's an interesting story to be told. But we'll wait for the last session because we need to lay some groundwork for this. Okay, now this is, I think, a couple of times I've forgotten to give outline or timeline, but this timeline will serve more or less as an outline, because the most of you who have had some science background recognize this man, Mendel, who did the work on peas. That was 1866. So that would be like, hopefully we can start from that era or a little before that and get up to the second stage. And this is the famous image of Crick and Watson, when they analyzed the structure of DNA. And the third stage was this one, when the Human Genome Project was sort of completed. It wasn't really completed until really this year. Absolutely, they didn't put the last word until earlier this year type thing. But they just thought, okay, well, we have enough, so we can go into the public. So this was, Francis Collins and the guy who challenged or lit the fire on the NIH, Greg Winter, who was the private entrepreneur. And so this was, they were, so that was the announcement, and the last but not the least, the two ladies who were awarded the Nobel Prize. So this is the outline. So now there are so many other stuff that one can say. We are not going to be able to cover a lot of spin-offs from this technology or science, such as in vitro fertilization in detail, stem cells, synthetic biology, a lot of advances in technology about sequencing, like from the old sequencing, it has gone to so-called next generation sequencing and third generation sequencing. And there is, of course, cloning.
Bharat, are you on your slides? Do you want your slides up?
Oh, I'm sorry. Yeah. Yeah, I think I'm not sharing. Sorry, that may be the part of the problem. I think I did not share. Or maybe I should. I was sharing, I thought. So just a second, let me double check. Aha, share screen. Yeah, I think now you should be able to see it. Now you can see the slides, right?
Yes, it's good. Very good.
Oh, okay. Oh, yeah. Sorry about that. Yeah, thank you. So, yeah, thanks for pointing it out, Peter. I saw that. Yeah. So, yeah, I think I had forgotten to click the share button. So I think I started the recording, but I forgot to hit the share button. So this is the thing that we're talking about. This is basically the outline as well as timeline, okay, of our class, okay, that we are going to be talking about from Gregory Mendel, the monk, to Crick and Watson, who deciphered the DNA structure, to Crick, the Human Genome Project, and the two ladies got the Nobel Prize. Okay, so this is, but we're not going to cover some of the other stuff, such as in vitro fertilizations, a lot of details of stem cell biology, and because I did some of that in the mini medical school class, so there was some overlap there. A lot of the stuff that is done with synthetic biology, which is a whole new discipline by itself, cloning, and then a fringe movement called transhumanism, where people do try to inject CRISPR themselves. I think there are some really, some outlandish work that is done by some people outside the scientific community. Now, some of the names, that were given, those that we'll get into the Mendel's history type of thing then. But the name of genetics was given by an English biologist in only several years after him, a guy named William Bateson. So he was, and there was study of inheritance, and that was the name that was given. And on the other hand, a Danish botanist named Wilhelm Johansen, he had coined the word gene to describe Mendelian units of heredity. And he also had, made distinction between the outward appearance of the individual, which is called phenotype, and the genetic traits, which is called genotype. And hopefully some of you who don't have science background did look through the glossary that I had sent out. And for those of you who are in the so-called classic education, know their Greek, and genos means birth, so that's how the name was coined. Okay, and now, so this is, and this was, this is an interesting, it's amazing how much information one can find in public domain that, as you may recall from high school biology, Mendel worked with peas, okay? And the reason why he worked with the peas, because initially he was working with rats, okay? But the monks did not think that the abbot of the monastery where he was stationed in what is currently Czechoslovakia, they did not think it was too kind for priests to be having animals indulge in sex, so he switched to a more benign target, the peas, and which worked out good for him because that was, and at least until 1990, finally somebody figured out the genetic cause of the wrinkled peas, because there is a gene that is abnormal inside the peas. Yes, peas do have genes, by the way. So, and that has to do with it causes it to lose more water, but the outside coat of the pea fails to shrink. That's why it looks wrinkled. So finally, it took almost more than 130 years to figure out, as to why that was happening. So finally, the so-called phenotype that he observed was correlated with genotype. So it's an interesting thing as to how science works. Okay, now, we can say the genetics is, branching out just like a lot of disciplines into varieties of branchings. The classic was the transmission genetics, which was basically observation. And then 1950 was the whole discipline turned to so-called molecular level, And then with the genome analysis, we are into population genetics. And now we can say we are into quantitative, with the massive computing data and the comparison of sequences to our going back to the early hominoid species, we can compare. So there are the varieties, and there are other branches, also that are coming into existence. So this is the way genetics is working, you know. And obviously there are other things called. Now I think this is wait, don't want to waste time. This was one of the Monty Python sketches on the meaning of life, basically. So okay, so you get the idea that we're always, as human beings, we've always been trying to figure out as to who we are, you know. Okay, and where did we come from, you know, which now who we are, you know, can answer in one word, you know, for those of us who remember Desmond Morris. Oh, we are finally naked ape, okay, or otherwise like baby chimp, you know, if we want to get to the hereditary things as to, you know, because when we compare, we are very similar to chimpanzees. But you know, we're the smaller baby version of chimp, so to speak. You know, so that's how we can say, where did we come from? You know, with due respect to all this ancestry craze, you know, I think we can say, well, simple one answer: we all came from Africa. Okay, so but we have always been curious about heredity. It's with the first time the words heredity became popular introduced in English language was like 1530. And at that time, because of the Holy Roman Empire, it was the idea was to condition of being an heir. So that was primarily related to property, and then with with the expulsion of the Jews and other people and the color, in Spain, it started. The blood was becoming more important, so a state was the blood heredity became into meaning because with all the inbreeding among the kings and royalties of Spanish Empire, of Holy Roman Empire, a lot of them were quite sickly, and they were not able to produce male heir. So that's when and there was a geneticist in Spain, because Spain was mighty empire in those days, and his name was, I think, Luis Mercado, and he had talked about heredity of the royal line. So the meaning is, it's fairly new, old type of thing. And then later on, obviously, heredity with the royal line, became a lot more important. And when it gets down to meaning of life, with all respect to, so I think, it more branches out into metaphysics and philosophy, okay, type of thing. But this is, for those of you who know their Greek, can probably translate this for us, okay. On the other hand, if you're taking some Latin, you may recognize this phrase, nos geti ipsum. And it's basically, I think in plain English, we would say, know thyself. So we've always been, and it's been used, from the classic literature to Thomas Hobbes in Leviathan. So we've all been anxious to know ourselves, type thing. So that search has been going on from the early days of living in caves to present day. And so in a lot of this search, actually on a more scientific basis started with this man, people recognize who this is. Okay, you can unmute and speak up type of thing if you want to, just to make it more interactive. Yes, okay. So I think that is Anton Leeuwenhoek, so he's there, and okay, and that is, contrary to the similarities, he's not Weird Al Yankovic. Okay, by the way, the hairstyle is very similar, but he devised, he was the first one who actually looked at and coined the word cell, so because it looked like the Jesuit monk cells, so that's how the name cell became, and then it became adopted to the medical terminology. Okay, so that was the start of the modern, pathology, and then later on it morphs into molecular biology. So in another 100 years or so. Okay, now the question is, do you really want to know yourself? Because this is the problem that can come up with. If you do your genetic analysis, I mean, this was one of the logo, at this of one of the new biotech companies, is to know thyself. But you really want insurance company to know that you're at a higher risk for coronary artery disease. But I think there are laws that the genetic information you cannot be denied. So I think there was been in the second Bush's era, the rules were passed, but still the information can percolate to your employers, and even though you may not be denied insurance, but I mean there can be some. So privacy becomes a big concern type of thing. So this is and at the same time, this is coronary artery disease is probably beneficial that you start looking after yourself. But certainly, I mean, the question comes up with, if you have a dominant disease which is untreatable, do you really want to know? I mean, because it would change your lifestyle. For example, like the breast cancer gene. Now there at least one can intervene. But if you are going to have early Alzheimer's, do you really want to know at that stage? So it becomes a very, very ethical and philosophical dilemma. Okay, and now this is, and any class on biology has to start with, the man who was, the grandfather of all. And this is the famous statement, which if you've taken other classes with me, I mean, I'm sure you recognize this. There is grandeur in the view of life. It's a beautiful piece of writing. This was the last paragraph in the first edition of Charles Darwin, and it was 24th November 1859. So, okay, and then later on he goes on to say that light will be thrown on the origin of man and his history, type of thing. So, okay, and again, there was, and now this one obviously he was fighting an uphill battle with another classical paleontologist such as Richard Owens, who is the man behind the Natural History Museum of London. And then also, if you remember from high school, Lamarck's, thing, the so-called giraffe theory. So, and the so-called soft inheritance ideas that were being popularized, and these were finally, put to rest by a German biologist who said, well, if there is a need, the organ would change. And he actually cut the tails of the mice. And obviously the seventh generation of mice still had tails. So it proved that there was, everything was not inherited. And even Darwin himself was puzzled because he tried to come to terms some idea with it, and then he proposed something called gemmules, means tiny particles, that were there which were populated in all the individual tissues, But obviously nothing was known at that time. So it was still, he was struggling with the concept, And then other people tried to say, well, can you transfer heredity by giving blood transfusion from one to the other? Didn't work out. So the idea would still remain on the shelf. And okay, and this is the man himself, at the Museum of Natural History. And there was a lot of, this thing with Richard Owens, who actually opened the museum to the public, because before that it was only allowed for the real scientists and the people who were at the proper contact, So he opened it to the general public, and but he did not like Darwin, so he put a statue in the back, and Richard Owens' statue is right in the front, by the way. And so this is the man himself, and this is the classic work that changed the whole idea of evolution, the origin of species. Okay, second book that he wrote, which got him into even more trouble with the church and the belief at that time, the descent of the man. And there were several cartoons in the English press about, the man descending from apes. So that was, that was the descent of the man. That was his idea, which was harder for people to swallow at that time. Now, he has written about, he was a very prolific writer, in spite of his whole problems. And this was one lesser known book that he wrote about heredity in other species, the animals and plants under domestication. Because gene editing has been around for a long time, since the early days of Fertile Valley. People have been doing, gene editing. Farmers have been doing that for a long time, and that's how domestication of animals started. Plants, and that's how we got all the other grasses. Okay, and the man now it blows my mind that this book was published sometime, like 1870 or 1880 type thing, but it traveled across the Atlantic and was. By the way, I think we'll get to that afterwards, type of thing. But does anybody recognize this man? Who this is? If you suppose, speak up, please. Okay, he's from Finland. Okay, if that is of any hint. All right. Okay, so I think well, this will make it clear. Now, do you know who that is? Okay. All right. This is Svante Paabo. He was the Nobel Prize winner for medicine for this year, and he is pioneered at the Max Planck Institute in Germany now, where he isolated Neanderthal DNA, and he's also gone on to create the full genome of Denisovans, the other human twig. So and this was several years ago. So this was finally he got his turn at the Nobel Prize. Now I showed you the other book, which is going back, The Descent of the Man. The variation of animals and other things. So now this is obviously you're not going to be able to recognize this person is, but if you are from California, then you may recognize that this was a guy named Luther Burbank. Does anybody who's into botany and horticulture does recognize that name? Okay, he was a self-taught botanist, horticulturalist, or a plant. So he himself has edited and strained about 800 different strains of fruit and plants and flowers, and he was inspired. He was not, even though he was from Massachusetts, he was not a Harvard graduate, or was a self-taught farmer who, initially, but his father died and he was left with his farm, and decided to, get really inspired by the book that he read, and made successful seeds and business, which is sold, and it moved to Burbank, California, and experimented with varieties of flowers and seeds. And the most famous one is this, russet potato. So, and he was well respected, not just in United States, but all over the world. In fact, the famous botanists from Europe, at that time, United States was sort of in the backwaters, and the famous botanist from, I think, Scandinavia, Hugo de Vries, came specially to America to meet this man, to see his work. And he had his own theories that he thought he was going to be the self-taught Darwin, but some of his ideas, actually, research was not very scientific, but he was amazingly popular, and he got funding from Carnegie Foundation, because Carnegie believed it's the American thing about the self-made man. So he got grants up to $10,000, several times. So, and the other book that I had mentioned in my reference that sent out earlier by Carl Zimmer, who writes in a different viewpoint. It's a more interest in history story type of thing. And the book's title was She Has Her Mother's Laugh. It's about the powers, perversions, and the potential of heredity. And Carl Zimmer is a very respected science journalist, So this is, the story of, how Darwin's influence extended all over the world. And even in China, that was, his books were translated. And so considering that this was before internet and telegraph was barely being invented, he had a worldwide influence. And unlike other cultures, the people in the East were a lot more willing to accept evolution as a concept. They did not have any problems with this. And so these are, we can divide the classic genetics. This is done from this slide is from the Core Spring Harbors Laboratory, which is a place which we'll figure widely in our talk as we go along. And so this is, this is not telling you anything that you don't know. Every parent knows that, oh yeah, she looks, like her mom or her dad. And, these are just basic principles that, that's there for completeness sake. And I already mentioned William Bateson's. And then again, Darwin was in trouble. So he had and this person is, these are some of the English cartoons. So Thomas Huxley, who was a scientist in his own right, and he was quote unquote titled Darwin's Bulldog. So he defended Darwin's theory, because Darwin was a very withdrawn, shy person. He had his own health problems. So Huxley went to bat for him, and he debated the famous clergyman Wilberforce, about him, and so it was a big this thing in the Victorian era. So okay, so this was a varieties of influences that Huxley popularized and defended Darwin's theory. And we already talked about William Bateson coining the word genetics, type of thing. And this was the earliest start of genetics at Cambridge Genetic Society. But the big problem that was that genetics is per se not deterministic, but more probabilistic, because it's a lot more complicated than what we realized, and that's the problem in turn with biology. And this is the cartoon of William Bateson that was made up in the English, of popular press. Okay, and this is, somebody had recently written a book about the Huxleys were like kind of the royalties of British scientific establishment. Okay, so Leonard Huxley, Julian Huxley was his son, and Aldous Huxley, who, you may have read the book in high school. Okay, the Brave New World book that was written. I should say probably, I'm not sure, but I would say probably in the 30s, that thing. So this is his plaque, of the English heritage. And somebody has recently written a book about them also. So I haven't read the book, but about the biography of all three, Okay. And now this is the classic genetics, just for completeness sake, we should go over that, and I already mentioned it. He switched from mice to peas. And this is the classic genetics, or something called the Punnett squares, that is outlined as to the dominant versus recessive, as to, that a big B versus small b. And, this is the symbol that is done on the right side. This is the female symbol. So if you have two genes and the dominant, so as to how dominant and recessive, so 3-to-1 ratio that would be produced, so that was the Mendel's laws, and we'll go over that. And this was an interesting picture that this is, you have to see, these were the monks at the Abbey, in Brno, presently Czechoslovakia, was part of the Austro-Hungarian Empire. And this is Mendel up in the corner. So he's staring at the flower. And you have to admire the style. Okay, this is like a contrapestal style. He's standing with, okay, with the Italian statue type of thing with his hand on his waist. Okay. So then, and again, it's a very interesting story that he came from a limited background, and just to get the free schooling, he decided to join the monastery, and he failed miserably as being a teacher, and several times he failed chemistry exam where he had to take a train to Vienna, and initially then he finally he found his passion in researching, the plants. And the reason for this research was, this has again, how economics winds down, because people were Central Europe was interested in breeding sheep at that time, because the wool export was this thing, and the Spaniards, monopolized the merino sheep. Okay, and they were almost illegal to export sheep from Spain, and then some of them were sent to England, and then the sheep industry in England took over, and then some were sent as a gift, to there was no Germany, but were sent to Prussia or something, and so people wanted to breed good quality wool because it was economically very, very profitable. So that was part of the reason. So that's why some of the Hungarian gentry, encouraged people into researching heredity, and that was one of the reasons why the Abbey was interested in doing this research. So some of the ideas where economics played a role. So he tested, the various, and he was lucky that he meticulously checked varieties of traits, with the ones that are obvious, the texture we talked about, color of the seed, the flower, the height of the plant. Okay, so, and he had read a lot of the other publications that were done in botanical journals at that time. And then this was the squares were named by an English professor at Cambridge named Punnett. So this is called Punnett squares, So the varieties of traits we carefully analyzed, and he must have read, thousands of plants. And at that time, Vienna was the center for the science and was the cutting edge. So he sent his report to a well-known botanist in Vienna named Carl Negeli, Naegeli, who had studied hawkweed, which was harder to breed. And he just said, okay, well, I mean, this doesn't make much sense. He didn't take much of this the monk in the provinces, So his work was and he had published his paper at a really less known society, basically. So the Proceedings of the Brno Natural Science Society. So obviously, so it didn't really make a big splash. And obviously it did not make it to Western Europe. So Darwin, he may have sent a copy to Darwin of his paper, but I don't think there is any mention in any of the Darwinian scholars that he actually ever read it. Okay, and this is again, so how the idea of the dominant and the recessive, as to which is the stronger state of dominant and recessive, that he clearly worked out the simple, not mathematics or arithmetic of this thing. So that was the start of genetics, of classical genetics as we've come to know it. And then he, based on that, he formulated certain laws, the so-called laws of segregation, law of independent assortment, and the law of dominance. So this is, so this was how he presented a copy of his paper to the Brno Natural Society. So it went unnoticed, as we mentioned a little while ago. So for unnoticed for a long time. Okay. Now at that time, obviously Carl Linnaeus, the classification was big into vogue, and he was a Swedish botanist, biologist, and he classified all the things that were tortured us through biology about the phylum and taxa and genus, and class. And so this was, and after classifying all the animal kingdom and the plant kingdom, he thought, well, it would be reasonable to apply the same laws to humans also. So this is, I mean, almost blows your mind as to when you look at it with our present, ideology as to how this was his classification of human. Okay, as to then you can just look at the color of I mean, this is almost mind blowing. The Americanus, red, black, straight, thick hair, stubborn, zealous, regulated by customs. Asiaticus, ruled by opinions, Africanus, okay, and that is, flat nose. I mean, this is unreal as to how the description was, but that was the state of the art. And the Europeanists, obviously, the white, blue eyes, gentle, inventive. So it was like, so that was his way of classifying. And we already talked about, so it, obviously, that was they're the product of their time. So perhaps we should not judge it too harshly, type thing. Now, when you talk about real biology or pathology as of disease, it started with the classical statement in biology. Yeah, okay. Yeah, thanks, Peter. Peter mentioned in the chat the Huxley's An Intimate History of Evolution hardcover. Yeah, they just came out on November 16th. So that's, yeah, so that's, it would be, there is so much to read, so little this thing. So I think, but I will get to it. And this is the famous statement, okay, all right, cell comes from a cell, basically. So that was the, famous statement of the grand man of pathology, Rudolf Virchow. Okay, Rudolf Ludwig Karl Virchow. He was born in 1821, died in 1902. And like a lot of Europeans at that time, he was a polymath. He was a physician, anthropologist, pathologist, historian, biologist, writer, editor, and politician. So he's rightly considered, like the father of modern pathology, as well as social medicine. And the varieties of terminologies that we learned in our second year of medical school, terms leukemia, i.e. white blood. And I mentioned that in the mini medical school class. And then that's initially they thought the blood was filled with pus. They didn't find infection, and that's when the label was given as leukemia. Then he coined varieties of terminologies such as embolism, that is the thrombosis, the blood that clots on the side, and embolism that travels, and varieties of other terminologies. He had amyloid degeneration, spina bifida, all these terminologies. But at the same time, he was not exactly sold on the Darwin's idea. Okay, and this was interesting that he believed that a lot of the illnesses were related to poor social conditions. So this was don't know what to make out of this statement, but perhaps that does make sense. Medicine is social science, and politics is nothing but medicine on a large scale. There, I'm still scratching my head as to see what he means by that. And this is classic example of your cell that you can see that. I think I can't see my magnifying lens, hopefully. Okay, yeah, I've got it here. Yeah, okay, yeah, now I've got it. So you can see this is the classic cartoon diagram of a cell, which is the cell wall on the outside and the nucleus, which is where the, and these are the two types of cells, with the so-called prokaryotes and eukaryotes. We are all eukaryotes because our DNA is important and is nicely preserved inside the nucleus. Okay. And on the other hand, the bacterial DNA is just lying around like a circle, basically. So this was, the classic diagram of a cell that we talked about. So this was the start of microscopic cellular biology as we have known it. And it's the state in vogue, and even now, there's some of the stains that they used because there was the early rise of, dye technology in Germany, and a lot of the stains that were used, are still used. The methylene blue stain and the, other testing, they're still used. If you go to the pathology lab, they're still used for a microscopic diagnosis. Now, again, like people, it took a while for some of the work for Mendel's work to be reproduced, by other botanists in Western and Central Europe. And so finally the idea was being accepted. And this was a physician named Frederick Michener, and he was a qualified physician, but he had trouble with his eyesight, so he became a researcher. And he was not aware, of Mendel's work, and he was researching sperm in salmon. Okay, and he was working on the idea of inheritance, basically, and then he collected, obviously, the microscopically, the new white blood cells. So collected white blood cells from pus of bandages. He lysed the cells with, dissolver and detergent, isolated the nuclei, and found a substance, which was present in inside the nuclei. So he labeled it as nuclein, and that was present in each test. And obviously, the chemistry was enough advanced at that time that this was high in phosphorus. So the early idea of heredity was, coming into vogue, by a guy named Frederick Michener. Okay, and this was again, at that time, obviously there was a whole concept that were going on where the end of vitalism theory, that was in vogue from the ancient times on. So that was put to rest by this time. And then later on, the German biochemist, they named Albert Kossel, who gave the name DNA, which identified nuclein as a nucleic acid. He isolated the nitrogen bases, which were considered to be the basic building blocks of DNA and RNA. So this was, so considering the time, the chemistry was, the chemists were right on the forefront, and the terminology that they gave, we're still using it. The consequences to which we'll talk at length, which are the building blocks of heredity: adenine, thiamine, cytosine, guanine. So this was, the names were given as far back as the late 19th century, I should say, or early 20th century. Okay, and this is, the basic. Every genetic textbook will have the classic pedigree. Okay, and pedigree, by the way, the word is French. It has to do with the feet of a crane. So pedigree. So this was, and this is, again, the hemophilia. Because with a lot of interbreeding, Queen Victoria had 9 children, including Beatrice, who was a carrier who introduced the gene to the Spanish royal family. Okay, and then again, it went spread all through the, royalty of Europe. And you can see, like this is, this is an old textbook. So I think, if you, unless you know your European royal history, I think, and it also went all the way down to the Tsarist Russia. Okay, and one of their sons had hemophilia, and he was, the attempts were made to treat him by the famous guy named Rasputin, as right, so that was, the history of heredity. And so this heredity has been around, and this was, and people were aware of blood groups, and even, heredity made its legal appearance into American juries, because the famous actor in Hollywood, Charles Chaplin, had a child outside wedlock, and he was sued for for alimony or parental contribution. And at that time, the first time it was used to prove that, yeah, he was not the father, because his blood group was O and the baby's blood group was B and the mother was A. So that was, so obviously he was not guilty, but he still, the court still ordered him, well, the baby looks like you. So they're all average Caucasians. So I think, he had to still pay the woman who had accused him. So, okay. So this was, okay. And so there I have so 10 of Victoria's male descendants, were afflicted with hemophilia. So, okay. So that was the so-called, we'll talk about the inheritance. It's like the sex-linked disease. So the women can carry it. And the genetics, for those of you who are not science major, how this coded in the square is for male and the circle is for female. And the afflicted is considered in dark squares, and the carrier is, written in half. So that's how the genetic thing about hereditary charts were formed, before the, molecular ways, So this is how the genetic charts were interpreted, just to give you a little bit more scientific insight into it. Okay, and this is, so this is chromosomes with the staining technology and other stuff. Chromosomes were diagnosed, in 1882 by a German scientist named Walter Fleming. And so he had a PhD in cytogenetics, and this was the earliest way of, now most of the chromosomes are not as neatly arranged and not color-coded like this, but this is just meant for illustration and explaining that they're of different sizes, like they can be, chromosome 1 is quite large. On the other hand, the male Y chromosome is tiny, puny little thing, at the corner type things. And you all know that, we have 23 pairs, while the chimpanzees have 24. And so this is, now this, people were aware of the chromosomes, but it wasn't, this was a woman, an American geneticist, named Nettie Stevens, who was, and she was the one who had actually isolated and identified sex chromosomes. Okay, and so, and initially, even they were wrong with the counting. People thought that that there were like 48, and it took until as late as 1950s, to say that the numbers were wrong, that they were 46 actually. Okay, and so there were other people, and so Nettie Stevens in 1905, described the X chromosome. Okay, I think so. And then different species of other ends have different numbers. And if there are abnormalities of these chromosomes, so if the part of that is broken and attached elsewhere, you can get the classical genetic abnormalities, including Down syndrome, which was the name that is no longer used anymore, which was called Mongolism in the old days, when I was in school. So that is the abnormalities of duplication of chromosome 21. And there are other rarer syndromes that I'll have some slides to follow in just a second. So this is just for people who have more scientific interest type of thing. So these are the varieties of Down syndrome, which is well known, which is the classical eye nose bridge that is breast. And it's interestingly, they have a single palmar crease. Okay, they have lack of muscle tone. And more importantly than that, they have some serious heart abnormalities. Okay, and nowadays, obviously, with the early diagnosis and amniocentesis that was available, and now it is the cell-free diagnosis for older women who are expectant mothers. It can be diagnosed very easily based on blood sample analysis. In the back in the 70s and 80s, people needed amniocentesis, which was a risky procedure to begin with. Okay, and there are other syndromes which fortunately are more rare. Which is the trisomy, means there are 3 these things. So gene duplication plays a big role also, and part of the chromosome can be broken. So there are varieties of abnormalities of chromosome structure itself. So you don't necessarily need the abnormalities of the genes, but so these are some of the features. And this is just given an idea about, you can see how the Y chromosome is small, the X, 2X. And that's just this explains basically, because in a disease that is one gene that is inherited from mom and dad, the males have only 1 copy. So that's why they're much more likely to show symptoms, So that is the example of the sex disease, So, okay. All right. And so this was a lot of the work was done by the German cytobiologist type thing. So, okay. All right. And there are now the numbers have been worked out as to how many base pairs are there on each chromosome, but that gets to be more than probably you would want to know. That okay, you mean missing such a small piece of DNA could turn a perfectly good human into a man? So that's I'm sure it was written by a woman geneticist, type of thing. So okay, and again, this is I know this probably makes more sense, type thing. We've messed up everything. So this is, so the evolution is not always. Now again, we're getting down to the nitty-gritty of it as to definition of what is a gene. So we can argue for that for all day, and you could just put a stretch of DNA that is transcribed into RNA that is transcribed into protein. So that's a very simplistic definition, type of thing. DNA segment that contributes to phenotype, means the external things and function. Okay, but doesn't, so there are varieties of definitions, and gets to be the more complicated definition that, is a locatable region of genomic sequence corresponding to unit of inheritance associated with regular. So that, and these are all these words have significance type of thing. So, okay, so this is, and the pioneer of American genetics is this person sitting well dressed at his laboratory studying with fruit flies. He was a geneticist born and raised in Kentucky and did most of his work at Columbia, named Thomas Morgan, okay, in the early 20th century. And they worked with fruit flies, and tried to isolate mutants, the males with white eyes, a red eye mutant, So this was meticulously, the fruit flies were easily breedable, and and the lifespan was short, and they were cheap to maintain. So that's why there was the organism. So we can study a lot from something called model organisms, and he left it wisely. And it was a motley bunch of crew that worked with him, so they were all characters by themselves, type of thing. And in fact, I think in 2016, there was a movie made about Morgan's Fly Lab. Okay. And that was, and there was one guy named Sturtevant that worked with him, another guy named Herman Mueller. And Herman Mueller went on to get his own Nobel Prize, and Morgan was later on he moved to Caltech, and he was also a Nobel winner, and he was still a little bit skeptical about in the initial phases about Darwin's theory of evolution, but he worked on and a lot of times based on linkage as to how the two mutations were associated in the same fly. His students, were able to establish something called genetic linkage. And in the classical days, because obviously, this was before molecular analysis, they were able to establish something called the centimorgan distance. So how the two genes traveled in pairs together. So there was some suspicion that this is not just a theoretical concept of heredity, but there actually was some physical basis for that. Obviously, remember, they're talking before they had the idea of, the structure of DNA and other stuff. So we talk about, but they were able to associate very characteristics and put, real physical evidence to that. Okay. All right, let's see what we have here. Okay, and again, for heredity, we're all worried about, okay, so we all look out like our parents. So heredity is cannot be avoided. So this is the inheritance. Again, we talked about the plants and peas. But again, it's much more relevant for human inheritance as well. Okay, so obviously, there are two lines of cell: the somatic line and the germ line. So the sperms. The germ line is gametes in the males, it's in sperms, and the females in the ovum. So these are called gametes, for the basic embryology or biology, gynecology that we talked about. And this is the basic conception that all of you are aware of. Okay, and so this was the way the chromosomal inheritance was established. The 2X and X, and X and X, XY. Okay, the parents were okay, and again, the similar things were worked out in Drosophila. Also, the chromosome pairs were thought to be very distinctive. And the males possess X and Y, and so this was the notation that was devised by the Morgan's lab. White flies with 2X were females, and along with that, the eye color was associated with on the X chromosome. Now the birds have it a little differently. Their chromosome pattern is labeled called W and Z in females and W and W in males. So birds are a little different, and perhaps because their inheritance pattern comes from dinosaurs, so it's a little different inheritance pattern type of thing. So okay, and again, some of the other mammals have a different chromosomal system. They have like 5 pairs of XY chromosome. So it's a lot more complicated, and nature has done a lot of experiments with varieties of species, through the evolutionary tree. Okay, and this is a little bit more complicated, type of thing. So I think we'll just skip that type of thing. So I think it is like, okay. And again, so now this was all well and good. This was in laboratory, but how does it get down to the real human health? And it went down to a very astute observer, who was a rare disease that he saw, but people got arthritis and other, systemic symptoms, which he had labeled as having, and the urine color was different because obviously in those days, blood tests were not that easily discovered. So they rely a lot more on urine. The urine stained of a different color type of thing. And he proposed that there was the diagnosis of alkeptone, which is anuria. And this was as far back as you can see how early it was, and he had associated that this was the condition that was inherited in the families. Okay, and so this was the first time he had linked a human health condition to genetic condition. So Archibald Garrett, so this was like the founding person. Okay, and this was before the word gene was coined. So you have to give hats off to the observation, that he put 2 and 2 together and came to a logical conclusion. Okay. Now, at the same time, now we'll get down into the dark history of genetics type of thing. And this was Darwin's cousin, Francis Galton. And in a few minutes, we'll take a break for. Yes, I think, yeah, just reading out Peter's comment, fruit fly genetics have all the fun. They give all the names, to seven less, son of seven, and other stuff. Yeah, right, exactly. So this is dumb and other stuff. And the geneticists they have the right to name. So there are all sorts of names that you can find, type of thing. So okay, and Francis Galton was a polymath, like he was interested in geology, anthropology, psychology, statistics. He's credited to have discovered fingertips, and he was also interested. This was time of Industrial Revolution, and there were a lot of social upheaval that was going on in Victorian England at that time. Okay, and so he coined the term eugenics, literally meaning good in birth, and he emphasized the so-called positive eugenics, that only the people who are smart, because he was big into, he was a great statistician. So, but look at all the judges, they're all born by smart people, and without putting an emphasis on the ability for resources type of thing. So he encouraged the whole science of eugenics. Okay. And he was widely traveled, and they established the whole discipline. And he received a wide support from the English intelligentsia. Okay. And the science of self-direction because obviously they saw that in animals and plants, okay. And this was the time when, they were able to access plants from the other areas and breed. And at the same time, the animal breeding for horses and sheep was also, much more in vogue. So that was the rise of eugenics, type of thing. So he thought, well, the same idea could be easily applied to humans. Okay. And again, but obviously that, we'll see in a few minutes, it didn't quite work out as well as he thought, Okay. And he had traveled to Africa, and he was, now again, at that time, the Victorian fashion was having, a dress was having a big bottom. And he had seen, what in those called, this is, I think, German, Namibia, or somewhere, I forget which area of Africa was it. So this was he says he could not take picture, but he took measurements of this African woman. So, and the word that they were used was Hottentot of Venus. So, and obviously thought, oh, this woman is naturally gifted, So, I mean, this Victorian women have to use this tons of padding to make themselves much more in vogue with the fashion. But this woman was naturally gifted. So he thought in a positive way type of thing. And again, at the same time, the one thing that is important that give him credit for in statistics, those of you who have taken statistics may remember he coined the term regression towards the mean. And which again, is very valuable for genetics as well. Okay. All right, I think we're coming up to 2:00, so we should take a few minutes break and type of thing, 5 minutes, and then probably reassemble in 5 minutes, because in case people want to take a water break or bathroom break or what have you. So I'll pause Zoom recording and we'll get started. People will filter in type of thing. So let me make sure that, okay, now I think I'm still sharing my screen. I hope people have this slide. Okay, yeah, so this is when we talk about Galton's type of thing. So this is, as to how he decided that, but it came up with, obviously, maybe there was a different intent, type thing. So, and his his ideology really took off. It was favored by a lot of who is who of English upper class society, including George Bernard Shaw, H.G. Wells. Okay, and so it was, amazingly popular type thing. And this was one of his statements, type thing, when the desired fullness of information shall be acquired from then, not till then, the fit movement to proclaim a jihad. Now this is, it's almost like, how many people had used the word jihad in, in 19th century? I mean, this is, or a holy war. So it's amazing how most Americans would not have heard the word jihad until 9/11, basically. So, I mean, type things like this. This is like written in 1905. And perhaps that may be because this may have to do with the, this fundamental struggle that took place in Africa, where the Mahdi was committing a hoary war. And I think the Brits, finally won. But I mean, there were some tough fighters. And even in 1857, Indian mutiny, jihad the word was around. So I'm sure it was in the British Empire, the word was more common than on the other side of the pond. Okay. And there are a lot of reasons for it, as we alluded to earlier, the industrialization, the people migrating to cities, So this was, became the scientific racism and the replacement theory, of existing population being replaced by the, undesirable classes, and obviously, that, and Americans, caught on to the idea very quickly and ran with it, so to speak, and outdid the Brits, as they've done in a lot of other disciplines, type of thing, like some of the areas that were started, and really they've, commercialized it, expanded it, and have researched it. So this was, again, and then this was funded by Rockefeller Foundation and other institutions, and the start of the Cold Spring Laboratory, in New York. And they were big into measuring, the size, because they thought, obviously, this was the idea of measuring people's skulls and fingerprinting, photography, and all those ideas being taken up. And again, the eugenic conference were held. The first major conference was held at the famous Cecil Hotel in London in 1912. Okay, and again, and in the United States also, the idea was carried out by a couple of psychologists and zoologists who got funding by the Rockefellers. Okay, I'm sorry. I'm going to skip the video of Thomas Morgan, because that is a long room, but and time-wise, otherwise I won't be able to get through this thing. So I'll send out the link. This is about his Morgan's fly experiments. But I've already alluded to it. This thing, but it's like a 2 or 3 minutes video. And this was a movie that somebody had made, which I saw it at USF in 2016. About the fly room, and as to how they succeeded just based on a meticulous observation. And this was hard work with growing flies with ripe bananas. So, okay, and so we, and the Morgan distance. And these are some of the axioms of biology. Is that worth repeating? That makes sense. Nothing in biology makes sense except in light of evolution. And what is true for E. coli is true for Jacques Monod. Was a later on Nobel laureate, and Theodosius Dobzhinsky was a Russian geneticist who did a lot of work at Caltech, later on. So, and mapped some of the, again, they worked with the fruit flies. So these were some of the famous statements. And okay, and so the basic idea is that was the genetic code that transferred information, which was carried out through generations. Now, obviously, at that time, the concept for energy was left to the side, and that again came into existence as to how the energy was for this mutation and biological changes was extracted from, from photosynthesis. Those ideas took much more later to get metabolized, and the role of mitochondria was defined. Okay, and then again, some of the genetics things were being worked out, by English bacteriology. Obviously this was before antibiotics, and the idea of experiments were done by a microbiologist, who worked with pneumonia strain, and they discovered that there were, just based on microscopic appearance, there were 2 types of strains. The colonies of bacteria that they could see were a rough strain, under microscope, and smooth strain. And the rough strain, when they were injected in the bacteria, the mice did not get sick. On the other hand, the smooth strain, they got sick. But then again, they injected the heat-killed smooth strain, okay, and the mouse lives. While the rough strain plus heat strain killed smooth strain, mouse died. So based on that, they saw there was something, inside the capsule that caused the death. So that might have been, so the bacteria itself was dead, but still the mouse had infection. So this was an idea that was proposed, something called the transforming principle that there was something inside the blood other than the, protein alone itself. So this were some of the earliest ideas as to how the genetic information was, and obviously they didn't, put two and two together, but this was proposed that they were capable. Genetic information was transferred to something called the transforming principles. Okay, and so this was the early days, and later on these experiments were much more solidified on this side of the Atlantic. Okay, and now this is interesting, about the politics. There's a British geneticist or a biologist, come a scientific communicator named Adam Rutherford. He has written several books on heredity, and this was his books about, his talk about called Politics of DNA. And this was the only Nazi memorial in London. Okay, this was the dog of the ambassador of German ambassador, who was buried, and he's still, he has his mark is Giro, the faithful companion, that's still in London somewhere. So next time you're there, you can look it up, type of thing. So, and okay, and this is some of the German propaganda, so that the Germans got on with this eugenics idea, and this was how some of their posters and the Nazis, got into in a big way, basically. So this persons with disability, you're carrying a burden that is costing the right 60,000 marks during his lifetime. So fellow Germans, this is your money. So this was the start of the official racial policy of the Germans, And then again, so varieties of of racial propaganda that was done. So how you're carrying the burden of the sick and the disabled type of thing. So, and then later on, it was much more formalized with the decree that Hitler sent out, and the so-called T4 killing propaganda. So, and that again, they went into it. So it's a well-known story type of thing. That okay, and at the same time in UK was not that far behind. Okay, this was in 1913, soon after the Genetic Conference, Mental Deficiency Act was passed. Okay, you can see, and like I said, varieties of people. This was an English side, Marie Stopes, who was the Birth Control Society proponent, and she was out and out racist, and talked about almost even as late as 1930s, wrote almost like a love letter and sent a bunch of poems to Hitler as to how he is trying to improve the German stock. So she was, she was even more outlier than a lot of the other people. Okay, and at the same time, even other famous writers such as D.H. Lawrence, he wanted to build lethal chambers to kill people who were disabled and not able to look after themselves, gently, so to speak. So, and some famous founders of statistics, Carl Pearson, Ronald Fisher, they were again strongly racist, not just against people with disabilities, the Jews, and so this was unreal as to how the land was taking around, And again, but at the same time, on the other side, there were other people who thought this whole idea was nonsense, including the famous writer named G.K. Chesterton. And these are some of the propaganda. Only the healthy seed must be sown. This was the eugenics in the British Empire. Okay, and obviously the Canadians and the other countries were not too far behind. So it was the whole idea had spread like wildfire through the British Empire. Okay, and this will come as a shock to people who to anybody in general. To me, this was news to me that the Honorable MP Winston Churchill proposed compulsory sterilization of the feeble-minded, and proposed using just a second. I'm going to check on something, please. Okay. Okay, good. Yeah, my recording symbol was not showing up, so but it is recording. So, all right, okay. So this was the idea that he proposed with this bill of mental deficiency that proposed compulsory sterilization of the feeble-minded, using Röntgen rays to sterilize men and women, compulsory sterilization in the mental. Okay, and he was far ahead of the others type of thing. Now, fortunately, this was opposed by G.K. Chesterton, who was a well-known writer at that time, and who lobbied another MP named Josiah Wedgwood. So eventually, the proposal by Winston was dropped. Otherwise, so it came very close to passing. On the other side, U.S. outdid them. That we went much further with these two gentlemen, from left to right, okay, a guy named Davenport, okay, and Goddard, who was a zoologist. Davenport was a psychologist, and he had traveled to France and had been introduced to the IQ test, okay, and which was the standard IQ test that was a Stanford Binet IQ test that was proposed, and he brought the copy of that along and tried to classify the so-called feeble-minded, and he gets the credit of coining the word moron, okay, and he went, to the extreme about, eugenics movement, and there was a school in New Jersey that was established, where for feeble-minded people, and some of the criteria that they used were just off the wall, so to speak, that based on appearance alone and pedigree, he was creating the pedigrees as to how it went around. And he was supported by various other who is who of the American establishment, including the famous, businessman Kellogg and other railway magnates, type of thing. And the whole idea, and Margaret Sanger, okay, the concept of better babies, okay, and compulsory sterilization laws were passed in 32 states, starting from Indiana. And the law stayed in the books until probably the early 70s, in some of the states. So in California alone, sterilized 20,000 people over the time. And Teddy Roosevelt was a big supporter of it, okay. And again, at the same time, so, and we talked about Kellogg, who was a big supporter. And it permeated the idea through the literature also in Great Gatsby. The hero Tom Buchanan makes a statement about the passing of the great race. Okay, and this almost blew my mind because I had to look up the word, type of thing, is to see when the word started. And this pamphlet was published in 1864. Okay, as was put out by the Democratic Society against Abraham Lincoln, as to how the Republican Party is trying to blend the races of the American white man and the Negro. Okay, so this was a hoax pamphlet that was put out at that time. Okay, and so this is, and obviously it wasn't true. It was the Democratic Party that was putting out, accusing the Republicans of this sort of, mixing of the races. Okay. And at the same time, there was a book written by a guy named Madison Grant about the passing of the great race. Okay, this was published in 1916, later on. So that had a huge impact on various people and was, it was translated to Germans, and, the Germans again, said, well, we're learning. And also Goddard, and they traveled to Germany and had almost given them a blueprint about using the Americans. So they modeled the Nazi Germany, modeled their ideas based on what the United States was doing. Okay, and so this is again, you can see that this goes around to the present thing, the replacement theory, the word still, very much used by Tucker Carlson. Okay, and even Elon Musk, is talking about, so the correct people are not having enough children, type of thing. So this is, so the idea 100 years plus years later is still very much in vogue. Okay, and this is a well-known story that is in all genetics books type of thing, about the Buck versus Bell. Carrie Buck was born in Virginia, and to Emma Buck, And again, so this was, and they were just based on appearance and some hocus pocus test, they were abandoned, type of thing. And they were institutionalized to Virginia State Colony for epileptics and feeble-minded. And then again, they were with foster parents, and was even though she was an average student type of thing, she got pregnant, after, as a result of being raped. So then she was committed to this colony for feeble-minded people and incorrigible behavior, promiscuity. And afterwards, again, so she was declared mentally incompetent to raise her child. So her foster adopted, and her baby was given up for adoption, and she was compulsorily sterilized. Okay, and the case went all the way to Supreme Court. Okay, and so this is just the map of the United States as to how many states had the mandatory laws of sterilization. So it's just, the numbers are staggering, to put it mildly, type of thing. So, and so it was, so US was the leader in international leader in eugenics type of thing. And the Third Reich, just, and so, and they sterilized as many as 400,000 people. Okay, so enough of the scratch past. So now again, so we talked about all the three main Western nations as to how the eugenic movements, and there has been the big dark shadow over the whole biology and genetics, which is we still are coming to terms with it, type of thing. So now, basically getting down to the real science, we talked about the inheritance and mutations. Now, we can talk about the varieties of genes, which cause, we can spend the whole 4 sessions about the individual, because there are as many as 6,000 to 7,000 single, inheritable diseases that have been, there is a whole category of mutational. It was written by a book, type of thing, by a first geneticist, who did the research on varieties of syndrome on islands of the coast of North Carolina, Dr. Victor McCusick. And but I think we can't just go around talking about that. But I'll just give you some examples of some of the diseases. Just some of the diseases, the achondroplasia, which is the dwarf status, and the other autosomal dominant disease, it is Huntington's disease, which is there's no official treatment for it, at least, and so that's the dominant disease. Doesn't matter what your lifestyle or what environmental factors are, you are doomed. You're going to get it, type of thing. Okay, so this is, but at the other same time, there are other diseases which are not so dominant, and you need 2 copies for that to show up. Like there's a huge inheritance of silent recessive gene of cystic fibrosis, sickle cell disease. The recessive trait may have stayed around because there is some benefit to protection against malaria, which is a well-known classical genetics finding. Okay, similarly for cystic fibrosis, may have given some protection against tuberculosis. So that's why the gene has not been eliminated by natural selection. Okay, so all right, so and there are other interesting rare diseases we can make a big case about. Like there are some cardiac diseases which are dominant. The Marfan syndrome, thick heart muscle, which is called heart myopathy. Which killed the basketball player, the Hank Gathers. So. And then again, the sex-linked disease, the classic example would be red-green color blindness. I'll have a slide for that afterwards. There's some interesting genetic link about that, so we'll talk a little bit more details about it, And then again, there are so-called polygenic diseases, which are, but when we had the hope that, when with the human genome, we'll be able to find the isolated genes for the particular common garden variety disease, such as diabetes. But that's not been as easy as we thought. Okay, and then it needs a huge database to come up with something called a polygenic score to get some handle on the chronic long-term diseases. Okay, and then again in cancer, we talked when we talk about the many medicals class, that the 2 hypotheses, that there is one gene that if you have inherited that is abnormal, then a spontaneous mutation that takes place that gets the cycle going for cancer. So that was the hypothesis that was proposed, but it's been proven with the eye cancer called retinoblastoma that occurs in children that, depending upon which eyes that are involved. So that is the two-hit hypothesis. So varieties of mutations that can take place, and there are numerous examples of it. Okay. And again, what does not kill you mutates and tries to kill you again. So we have seen that with viruses, type of thing. So, okay. Now, this is an interesting, medical mystery tape. Okay. The famous novelist Pearl Buck wrote a little lesser known book, The Child Who Never Grew. And this was known because she spent most of her adult life in China, and she had a child at that time, and she then developed and had mental retardation, and she wasn't able to look after her, brought her to the U.S. and was institutionalized. And then she wrote a memoir that I don't know exactly when it was published, but this was the book that was written. Now, at the same time, in 1930s, there was a woman in Sweden who had two children, was at young age, at 3 and 5, had mental retardation. Okay, and then she just had this. The kids were not just retarded, but they had a musty odor to their, their sweat, their urine, their skin, and even their hair. Okay, so this was a puzzle. So obviously, as a concerned mother, she was frustrated. She had no idea for it. She finally went to a doctor who was more curious and compassionate both, that to look at them and do some tests. So I will not try to pronounce this name, but obviously you can see Dr. Poling. That's probably the best way I can say. And see, he decided to take on some challenge. And obviously this would be took in 1930s, so the testing was relatively primitive, but he did the urine testing for looking for diabetes and infections and things like that. And he was also interested in chemistry. So he decided to do some tests for chemistry and did a urine test. Okay. And the urine test was like this. And for the chemist in the crowd, you can see that this was the urine turned green. Okay. And so lo and behold, so then he did further chemical analysis with either by himself, and finally figured out that there was an abnormality of one of the amino acids metabolism, and that was the start of phenylalanine, which is one of the essential amino acids. And you can see this was the start of. And obviously, his report traveled to UK to a psychiatrist named Dr. Lionel Penrose. And those of you who are Brits and scientists may recognize that the Penrose is a famous name for who is who, because his son is Sir Roger Penrose, who got the Nobel Prize a few years ago in physics for his work with, black holes with Stephen Hawking. But we are digressing. So he decided to do the same test in a mental institution hospitals in UK. Okay, and again found as many as 6 out of 340 were positive for this. So they realized that this was a metabolic disease and not just general mental retardation due to brain abnormalities. So this is almost like a medical mystery tale as to how. And then obviously it came to treatment, as it was almost a little too late for Mrs. Eklund. Okay, but then Dr. Penrose talked to other scientists, which was name was Hopkins, who was discovered vitamins, and that tried to devise a diet without phenylalanine, okay, which was just plainly carbs and fats type of thing. But the diet was obviously, we were talking about pre-war Britain, and it was expensive. It would almost cost like £1,000 for one child. So there was not, and it took until after the war when finally, for starving children, purified casein, milk protein was this thing, because when they started giving just carb diets, the children's muscles started losing their own muscles. So the phenylalanine test was again positive. So they had to come up with the correct diet, and it took several years with some clever dietitians' work, to come up with the appropriate diet for phenylalanine. And that's how the test came about. Okay. So that was the story of phenylalanine, and that test is obviously nowadays universally done for all newborns. So just thought it was an amazing human interest story of a concerned mom and a doctor who was curious enough to pursue the matter. Okay, now getting back to another story of this is the British Empire, okay, which is this was the start of the empire, and this is around here. Let's see if I can get down to a place called Calicut, okay. And in the early days, before the Industrial Revolution, cloth was exported from India to Britain, and that's how the name Calicut came about, and it was the name for calico, okay. I mean, you've all heard the word calico, okay. And now why does calico is important? Because in genetics, there's something called a calico cat. Okay, so I was curious to see why the name calico came about. Okay, I mean, what does a cloth material has to do with this thing? So this has to do with the how how in the chromosome the X, because otherwise during gametogenesis you get double copies of X chromosome. So one part is inactivated, okay, and so that's how the cat gets this calico stripping. And this was again applied not just in cats, but also was diagnosed by just on microscopy by a British geneticist named Mary Lyon, okay, and again, and also another Canadian scientist named Marie Barr. And so they had discovered the inactive X chromosome in a female regular cell. So the part was, so the females been trying to shut up men for a long time, and successfully, I may say. It's rendered inactive in a process called lionization. Lionization was named after Mary Lyon, who had discovered the process. The sex is determined by the presence of fire. And this was used for, in the early days in the Olympics, this was used as the buccal body analysis to determine true sex of female athletes. I think so. This is no this thing. So it takes down to the name, tracks down to the early days of empire. Okay, so an interesting. And this is the microscopic appearance of the inactivated X and active X. So okay, so this is how the microscopic appearance is. So this is a digression on as to how during fertilization the X chromosome is acted upon. Okay. Now the second era of genetics starts a little later, because by this time in the 30s okay, there was fear of science was coming along with the rise of the advances in science that were taking place starting from the classic book of Frankenstein, and I'm sure a lot of you would know that the Frankenstein was inspired by the experiments that were done with the early days of electricity, and she witnessed how the electricity was used for frog experiments, and the early days, even some cowboys had even shocked an elephant with electricity, in the early days of fight between AC and DC, and that was the inspiration for Frankenstein. And some of the science fiction writers were ahead of that, and H.G. Wells had written a book called Food for Gods, okay, which was about the people eating modified food and becoming gigantic, and so it was one of the dystopian type of novels. And later on, I already alluded about the Brave New World and various pulp fiction novels, which were talking about the perils that could occur with genetic engineering. Okay, and again, some of these things. Then after the 1940s, with the Manhattan Project, and the fears were even further aggravated of science that took place, not just of atomic science, but also of genetic science. And the science fiction writers were ahead, that they had coined the word genetic engineering in their literature before the scientists had any idea. And again, some of these ideas were, well summarized, with the Rachel Carson's book, and the book that was written in 1968 called The Population Bomb, was, and so that again caused a lot of fear. And at the same time, an English science fiction writer had written a book called The Biological Time Bomb, where he had hypothesized all the evil things that can occur with genetic engineering type thing. And this was the early days of, this was Time Magazine Scientist of the Year, okay? And this was US scientist, the name at 1960 Time Magazine, obviously was very popular in those days. And this was the earlier, after Dwight Eisenhower and between Kennedy, US scientists were recognized. And most of the names you can recognize, that most of them were physicists named Linus Pauling, Isidore Rabi, Emilio Segre, who was the atomic scientist, William Shockley, okay, who discovered the transistor, Edward Teller, the father of the H-bomb, Charles Towns, discovered the laser, okay, Van Allen, radiation belt, okay. But there were, in the small select group, there were two biologists, George Beadle and Joshua Lederberg. Okay, so the two biologists, were also, okay, and this was the fear as to whether the science has a power to play God or evil. Okay, so this was the science, and the title of this thing was Science at the Apogee of Power for Its Good or Evil. So in the early 60s, the groundwork was being laid out for distrusting science. Already you can see that that was happening. And one can understand as to all the horrors that we had seen with the atomic project and the fear of the Cold War. So it was stage was right, and things dramatically changed. Okay, and then again, so we already said about the early work that was done by Griffith, which was again, Griffith was shy and did not publish right away, and his work was kind of put on the side, with the war at the same time. But there was a physician in New York at Columbia named Dr. I forget his name, Avery, yeah, and Oswald Avery, okay. And he had repeated some of his experiments again, and he did it in a lot more systematic way about dissolving all the cell wall as well as the proteins, and finally establishing once and for all that with that the hereditary material was in fact DNA and not protein. Because at that time, people thought DNA was a relatively simple molecule and was unlikely to be of that important. Okay? And there were a lot of the biochemists and even geneticist camps were divided as to what was the hereditary principle. And people didn't think that that simple molecule with just simple chain of four components could be that important. So, but Oswald Avery, along with assistant from two other physicians who were Canadian and had worked with him, established without a doubt that, and then later experiments were repeated in a lot more sophisticated ways by a couple of other scientists, who labeled the isotopes phosphorus with radioactive phosphorus and sulfur with radioactive sulfur, and conclusively proved by doing experiment with something called phage. Phage means eating, okay? And these are the tiny particles which attack the bacteria, okay? So one can say they're kind of viruses that affect the bacteria, type of thing. So I'll have some interesting material to share about for phages later on. But so this was, and Oswald Avery's work was somehow, at the same time, the politics of science comes into the way, that he was nominated for Nobel Prize several times. But one chemist from Sweden who was in the committee, he was a strong believer that proteins where the action was like. So he had continued to block Avery's nomination. So Oswald Avery, then he retired and he was denied the existence. So his name is lost, compared to Watson and Crick, who established DNA as the principle of inheritance. So that was how one person's power on the Nobel Committee, and Okay, so these were some of the underlying history, which makes it a human interesting story. Okay, and on the other side, there were some other pioneering woman who was working, with maize, which was not that much interesting to other people, and she had done a genetic analysis and proposed something that the genes are actually mobile and they can move from one side to the other by looking at the various maize growth. And she had come up with the idea, this is so-called quote unquote jumping genes or transposable elements and so this was made from very quickly the genetic changes took place. And she had proposed this idea as early as 1940s. But again, first of all, this was a radical concept and it was, proposed by a female scientist. So her work was just ignored by the large community in large. And she, continued working on her own at Cold Spring Harbor Laboratory. And it wasn't until other people duplicated her work in 1983, when she was 81. She's probably one of the few single Nobel winners in her category, Barbara McClintock. So that's a name worth knowing. Okay. And so again, this is transposable elements of gene which jump from one area to the other. Now with the maize genome has as much of 90% of it is so-called jumping genes. Even in human genome, it's even a larger percentage than this is called retrotransposons. A lot of them are DNA, some of them are RNA, I think so. Okay. And so it has gotten a lot more complicated than what we realize. It's not just simple DNA and RNA that we talk about type of thing. So, and in other words, these the class 1 transposable element do not encode, but they produce RNA transcript, and they rely on various other enzymes, to transcribe RNA sequences. Okay, all right. Okay. And this was that famous, we threw out the name of Beadle and Tatum, okay, and they were biochemists par excellence, and they work with even a smaller organism, Neurospora, which is the bread mold, okay, and they created varieties of mutants and systematically eliminated, while growing the fungus mold, they eliminated the culture medium varieties of nutrients systematically one by one, and they showed as to how genes were responsible for enzymes, and they created the famous axiom in biochemistry, which is one gene is responsible for one enzyme. So that is how they were able to connect the so-called molecular technology with biochemistry. And so this is the classic statement that was recognized by Beadle and Tatum as to how genes were directly responsible for causing the workhorse in our body chemistry or physiology, called enzymes, which catalyze all the reactions that takes place. So by supplying varieties of compound and seeing as to what mutant strains develop or not, they came to a very logical conclusion that the genes direct the formation of enzymes. Okay, and so so this was a groundbreaking linkage between two major disciplines. Okay, and then at the same time, Irving Schrödinger, who's well known to physics aficionados, okay, that he was one of the quantum physics scientists, okay, who was with the famous Schrödinger's cat, and when after leaving or being exiled from Germany after Nazi Germany's takeover, a lot of the physicists were heading out to Britain and United States, and he had now at that same time, they were interested in combining the various disciplines of physics, chemistry, and biology. So this was the famous speech that he had given as to what is life, as to how the hereditary material is, probably a protein, and he had coined the term, which is called aperiodic crystal. Now, even chemistry majors would have a hard time explaining, because sometimes the crystal can reproduce itself, and when they do not maintain the lattice formation. And that's where the term aperiodic crystal was coined by him. And he thought that this might be the way that the code of life may have been, that may be functioning. So this was, so this is how he was combining the two disciplines. Because by this time, the quantum physics was being worked out. And so they were thinking as to how some of the basic sciences can play a role in, explaining the origin of life. And on the other side, the control systems were proposed by combining the early computing science mathematics by a guy, some of you were in college back in the early 60s, may have even heard about the cybernetics. And so this was science of control systems, communications, math, and biology and machines. So this was the discipline called cybernetics. And this was a very popular thing back from 1950 onwards to the early 70s, okay. And it went through so he had written several books, but this was one of the most popular ones. And let's see, what else do I have? Oh, sorry. I think that I had a picture of his book, but his book's title was "What Is Life?" And that influenced. I think somehow I didn't populate type of thing. So, okay. Now, it convinced a lot of physicists, including the famous James Watson, to switch disciplines, and got attracted towards biology. And not only just Watson, but Francis Crick, who started out as a physicist during the Second World War. So he switched to biology. A lot of other physicists, who were from South Africa and US, were started working on other biological disciplines, working with phage, okay, which is the tiny virus-like particles that affect bacteria. And so switch disciplines. So his book had an impact which affected a life of variety of science pioneers. Okay. Okay, so let's see. And so they all. Okay, I'm sorry, some of my links are missing. Okay, now we'll move on to this. We're 10 minutes, so we're right on time. We're coming up to what originally I had thought I should get through. Okay, and so this were, now Watson was, prodigy, and he had finished his early PhD, and at Purdue, or I think Purdue, yeah, not Indiana, no. And then he was doing postdoc and initially in Denmark, and he wasn't too happy with the work that was doing, and he had traveled to Italy for vacation, and he had listened to the lecture on X-ray crystallography given by the famous English crystallographer named Maurice Wilkins, who was working on biology, and at that time the molecular biology discipline was established at Cambridge. So when Watson listened to this lecture, then he decided to switch to working on DNA, okay, and asked for postdoc fellowship at molecular biology MRC Research Council in Cambridge, and he was accepted by the Brits. And then he was teamed up with another physicist. Watson was like barely 23, and Francis Crick was 35, and they were paired together. And they were not, exactly, they got along famously with each other, but were kind of looked down upon because they were loud and they had a lot of ideas, but not really a lot of practical work that was being done under their auspices. So they were, put elevated to the basement. And again, they were working on the model of trying to create X-ray crystallography model. Okay, and then they had the famous help from Maurice Wilkins, who worked with, let's see, let me jump up. Yeah, with this lady, okay, the quote unquote, who was an X-ray crystallographer of par excellence, and but was a strong-willed personality, being a woman in early days of science that didn't get along with. And when she was placed to work, under Morris Wilkins, she was under the impression that she was independent researcher, and those two did not get along. And this was her work that she had, taken some clear-cut X-ray photographs of DNA with X-ray crystallography, and how simple mounting works that they had done. You can see how this was just a simple pin on which they had mounted the DNA and was able to take X-ray crystallography. And DNA comes in varieties of forms, so-called wet and dry. And so she was able to get the real clear picture of DNA, which showed the classical double helix. And by this time, she was almost giving up on the work. And Morris Wilkins, being friend with Watson, showed her the photograph, and that inspired, Watson to work to build a model of matching all the A and T together along with G and C. Now, they were under pressure because, again, politics and DNA played a role, because at the same time, the giant of biochemistry, Linus Pauling, was working on it as well. Linus Pauling was at that time at Caltech in the United States, okay, and and he was a giant of protein chemistry. He had discovered the structure of hemoglobin, and he was working on it. So Watson and Crick had got the idea that he was working on it. In fact, he had written a paper, and this sounds interesting as to see. So this was, believe it or not, even though this was a basic chemistry method, the paper was accepted and published. And when Watson and Crick showed the paper to a real more qualified chemist, they showed that how the giant of chemistry had made a basic mistake. So then that just gave them the impetus to work even faster on their model. And now at that time, unfortunately to be fair to Linus Pauling, he was not allowed to travel outside the country after the war because he was a pacifist and he opposed the nuclear policy of the United States. So he was not given a visa. So he was never not allowed to travel to UK. And if he had traveled, obviously he would have had access to Morris Wilkins' work, and that he would have come up with the correct model. But at the same time, his personal politics came in his way, and that gave a leg up to Watson and Crick. And Watson and Crick were considered, you saw, I don't know if if you saw that they were so disliked that they were labeled WC by the other Cambridge establishment. So you can see how they were kind of not, they were pain in the butt, to put it mildly. And Francis Crick, when he discovered this, he wrote a letter to his son, as it says, we may have discovered the secret. And this letter was sold recently for $5.5 million for some collectors. Okay, so this was, this was the interesting story about politics, DNA. And Rosalind Franklin, by that time had moved on to different work, to a different college. And so there is a lot of controversy that people who are, think obviously, her work was stolen by that time. But again, she was working under Morris Wilkins, and she had already given up on working on this thing and moved on to a different project at a different college. So we can see where the truth really lies. And this was again dramatized in a movie starring Photograph 51, in a play starring Nicole Kidman. I don't know if it was on Broadway or off Broadway, but anyway, so that was a big scientific story type of thing. So, okay. And these are some of the famous papers, that they wrote, April 25th, 1953, with the molecular structure of nucleic acid, and you can just see that this was Watson and Crick came out from the molecular MRC lab in UK. And the second paper that followed a few days later, it showed that therefore likely that precise sequence of bases. And this was even more important paper because the genetic implications of the structure of DNA. And so even though, and this was the famous statement that they made, it has not escaped our notice that the specific pairing we have postulated suggests a possible copying mechanism for genetic material. It's obviously, that's an important statement, probably understated. But at the same time, this statement is even more important, that the pairing actually creates a code, because at the 3 stages, there are 3 important things: determining the sequence of DNA, and then deciding the code as to how the proteins are created or synthesized, and the information that it carried. So all 3 things are equally important type thing. So, but they had proposed both their ideas, and that started a race later on that we'll talk, probably next week, because, okay. And I'll just show you this film type of thing, I think, because we still have a few minutes, so we'll get it out of the way. This is DNA in its full glory. Oh, I'm sorry, our Alexa had started. Oops, I think, I'm sorry, I recorded my wife's voice. So you get the idea that A pairs with T and G with C. So, okay, so basically, so this is written in more understandable form. Organic chemistry that calls structure is function. Base pair determines the chemistry, A with T. So this is the standard, this thing. So, and these are quite tiny. The base pair is 0.34 nanometers. So you can just see it's quite small, basically. But still, when you look at the DNA itself, it's almost 2 meters long. So that's more than 6 feet. So it's wrapped around like a tight spoon, like your woolen this thing, or around proteins called histones. Okay. And so this is where the flow of information code is, stored inside each and every cell. Okay. The nature of the chemical bond. Okay. And where is this building? The Brits in the crowd, can anybody name that building? Okay, that is King's College, Cambridge. Okay. And this is the famous pub, Eagle a pub, where they came and announced that we have discovered the secret of life. And this is the plaque outside, the historic plaque, which you see all over London. In 1953, for decades, the Eagle pub was nearby the famous Cavendish lab. Francis Crick announced their discovery of DNA. Okay. So this is, the greatest scientific discovery of 21st century. Okay. All right. I think we are gone past 2 o'clock, so we should stop here. I will stop the recording, and then we'll continue the tale next week.
Fantastic introduction, Bharat. That was really super.
Thanks,
Okay,
thanks,
thank-
Brett.
Okay. Hopefully, that wasn't too.
You recorded
heavy.
the whole thing, so you'll send out the recording?
Yes, yeah, I did. Yeah, I did
Perfect,
record it. Yeah, so
yeah.
I figured out. So I
Well,
figured out.
good.
Okay, I think, so it will, because otherwise it's too heavy at this thing. And I'll send out the slides also, Unfortunately, I can't send out the videos because of the limit of 20 megabytes, so.
Not a
that's-
problem. Okay,
yeah,
thanks.
So, but the slides will be there, so. Okay, and I will, and if there is any glitches or any other
Thank you.
things, comments, please
Thank
jump
you.
in. Yeah. Okay, you're welcome.
Thank you very much.
Very
See you
good.
next week.
Bye.
John.
Thanks again, everybody.
That was good.
He's a hassle.
You're not on mute, are you?
Okay, I'll end the meeting, Joe, okay?
Okay, go ahead.